I can measure, take a single lysosome, I should be able to tell both the value of pH as well as the value of chloride for that lysosome. And so I'm just showing you the fact that you can take a pH sensor and stick it on to the chloride sensor. Just to tell you how the pH sensor works, if you make the solution a little acidic, this region is pH sensitive, it folds up and it changes fluorescence. So we now have 3 colors to look at. This green to red ratio, that's going to tell you chloride, and the orange to red ratio here, I'm going to call this donor to acceptor ratio here. This D to A ratio is going to give you pH. And so we were able to show that the pH sensing module is independent of the chloride sensing module. Regardless of whatever chloride value you have, your pH sensitivity of your device is retained. And the same is true for chloride. Regardless of whatever pH value you have, your chloride capacity, your chloride performance is the same. This is what I've just described to you. And so now you take this sensor and label lysosomes from skin biopsies derived from healthy individuals. This happens to be a fibroblast from Maulik in the lab, who there's no picture of him. I assume he's healthy. And so what we've done is, you see here 600 data points for 600 lysosomes. So if you take any data point here, you can say, okay, this lysosome here has a pH value of so much and a chloride value of so much. And so you just look at the spread, right? It's very hard to see what this looks like, so we've converted it into a density plot. And so this density plot really shows you that your highest value, the most represented values are here, the least represented values are in blue. And what we're able to immediately see is that there are actually 2 populations. There are 2 maxima, one here and one here. And so we were quite intrigued and we said, okay, does this happen for everybody? Is it just like one? So there's this one high chloride population lysosomes here and low chloride populations here. Okay. And so when we looked at 3 healthy individuals, we found that all of them had this high chloride population. So then what we did was we said, okay, let's take skin biopsies from a repository where these people were suffering, had been diagnosed with different subtypes of a neurodegenerative disease called Niemann-Pick's disease. Niemann-Pick's disease comes in 3 flavors, A, B, and C. And so in all of these, you will see that this high chloride population is lost. And in fact, in many cases, the distribution becomes almost a monotonous distribution. It's not this 2 populations. And so it really meant that there was something, the stomach seems to be this high chloride population, right? And so we said, okay, let's try and see if we treat them with therapeutic, what happens. And so Niemann-Pick A and B result because there's an enzyme called SMPD1 or acid sphingomyelinase that has been mutated. And so it comes in 2 flavors. One is a very severe mutation where you have less than 5% activity, and a less severe mutation which has more than 10% activity. So the second one is called B, the severe mutation is called A. And the therapy for this is to actually give the person the enzyme. You eat the enzyme, right? This is called enzyme replacement therapy. And so when you treat the cell with the enzyme, recombinant enzyme, you see that this high chloride population has come back for Niemann-Pick A and Niemann-Pick B diseases. Niemann-Pick C is due to cholesterol transport. There is no therapeutic for this, okay? And many times these children are given, they think that these kids have Niemann-Pick A-B disease, and they actually have Niemann-Pick C. So they will give you recombinant acid sphingomyelinase, frequently misdiagnosed, frequently mistreated, right? And you will see that many of them won't respond. But there is one very bad therapeutic that is right now being put through clinical trials. It's ongoing at the NIH. and you have very little responsivity. So here you see 3 patients. This guy has responded somewhat. This guy, this is a non-responder. And this guy also has responded a little bit. Why is this important? Because if I am so unfortunate as to have Niemann-Pick A or B disease, or Niemann-Pick C disease, if I'm treated with recombinant acid sphingomyelinase, it takes about a year before I see anything, before I see any effect. And it costs $250,000 a year, right? That is the cost of the therapy. And so it would be great to know beforehand whether I need to be on this therapeutic or not. And so then the question is clearly that high chloride population is doing something. What is it doing, right? What is the role of chloride? So the lysosome is actually a bag of calcium. It's called an internal calcium store. And one of the ways by which it functions is to be able to release its calcium in a very targeted way, right? And this is one of the main functions of the lysosome. And so we thought, okay, what does it do in terms of the capacity of the lysosome to release calcium? And so what you do is, the lysosome is anyway a balloon. So what people basically do is they puncture the lysosome with a chemical that causes a particular channel to open, and then calcium just sort of rushes out. And you can look at calcium rushing out by putting fluorescent dyes in the cytosol of the cell. And you will see that for a normal lysosome, you have whoosh, a huge amount of calcium comes out. But when you deliberately reduce chloride and you then puncture this balloon, you find that there's very little calcium that comes out, right? So it affects the lysosome's capacity to release calcium, right? Note here that we could not actually see whether the lysosome's capacity to intake calcium was affected, right? And I will tell you why we couldn't do that. Technology doesn't exist. So what people do is that they look at how much calcium is released, right? If I want to know how much air is inside this balloon, I measure how much air this balloon can release. Yeah? Okay. So the second thing that we asked was, well, we also know that the lysosome's capacity to eat is compromised, but why, right? And so here's proof of that. So we've marked the lysosomes with 2 chemicals. One is a chemical called lysotracker, which goes and gets accumulated in the lysosome because the lysosome is a highly acidic environment. So if you try and puncture this lysosome, or you affect lysosomal integrity, what happens is that the lysosome will bleed protons. And so then what happens is the lysosome becomes less acidic. And when it becomes less acidic, lysotracker will not stain it. The fluorescence intensity of this red will go down. On the other hand, there's a chemical called dextran, which is a polymer that's going to be sitting inside, and it's fluorescent all the time. So even if the lysosome loses integrity, you'll still see that the lysosome is there. It's just not doesn't have very good integrity. So if your lysosome chews up a substrate, so you've labeled the lysosome with both these chemicals, because you have red and green there, it's going to look yellow. So that's what you'll see. And now you add a chemical called GPN. And so enzymes on the lysosomes will cut GPN, and what GPN does is it punctures the lysosome and causes holes in the lysosome membrane so that it loses integrity. And then what happens is lysotracker will leave, and now you will see only green if everything is okay with the lysosome. And what we showed was that if you reduce chloride, what happens is that the capacity of the enzymes that are present inside the lysosome to function are compromised. This happens to certain enzymes, and it's not across the board. Only some enzymes are chloride sensitive. And that was our basic finding. So chloride basically does 2 things. One, it's really important for lysosomal calcium release, which is important for the lysosome to fuse. And the second is, it's necessary for the activity of specific enzymes that are present inside the lysosome. And now, when we were doing this, yes?
So were the identities of these enzymes known beforehand, that they are chloride sensitive through biochemistry?
One of them was known. And the others we just scanned through to see, because we know that the assay works and we had a positive control. And so aryl sulfatase B was another enzyme that we found. Certain cathepsins, but not all cathepsins. Okay, and so when we were looking at this, we found that there was no way to measure calcium inside the lysosome, right? And then we said, well, why is this the case? This seems to be very fundamental. Are biologists actually happy with this, that they can't measure calcium inside the lysosome? And it turns out that they were very furious for the last 30 years and very frustrated that they couldn't measure calcium inside lysosomes. Many people told me that please solve this problem. So we sat and thought about it, and that's what I want to talk to you about. We've just managed to solve a 30-year-old problem. And I think quite a few lysosome biologists are quite excited by this. But I want to tell you what the problem is. Extracellular calcium is about millimolar. Inside the cell, in the cytosol, calcium is nanomolar. And this 10,000-fold difference in calcium is what drives all of biology. And the cell signals to itself by elevating calcium inside the cytosol to tell the cell to do a job. The master regulator that decides to do this is an organelle called the ER, which is really a bank of calcium, which is also millimolar concentration. And what it does is it opens channels that releases calcium, and this tells the cell, excites the cell, and sets off signaling. But again, this is not done by itself, right? I can't simply randomly, if the ER opens, then we'll all be dead. And so you have what are called internal stores of calcium. So these are acidic stores of calcium, little bags like the lysosome, the Golgi, and the mitochondria. All of them have small levels of calcium that they generally release small puffs of calcium that then causes calcium to be released. And so we've always wanted to look at these internal stores of calcium but could never do so. And so what I'm going to talk to you about is give you some examples of taking one organelle and one protein on one organelle. So whatever I'm saying, just multiply everything by 10. I'll give you 3 examples of how this happens. So for example, if you take this protein called TPC, the 2-pore channel, which is on the lysosome, which is one of the acidic stores of calcium, when its ligand binds, TPC will open, release a puff of calcium, and this causes the lysosome to, this causes the channels on the plasma membrane to open, which then changes the membrane potential, and this is really important for insulin secretion. In another flavor, for any cargo to go down the endolysosomal pathway, you need to have normal trafficking that happens. So when early endosome and a late endosome or a lysosome and a late endosome dock, again, when the ligand binds to TPC, it releases a puff of calcium and these 2 compartments fuse. Lastly, the ER the lysosome can actually go close to the ER. When its ligand binds, it triggers a puff of calcium, and this causes the ER to then release its bank load of calcium. This is called calcium-induced calcium release, which is also extremely important. But none of these processes, you actually want to be able to sit inside the lysosome and watch the calcium change, which we've never been able to do. And why is that? Because every single calcium dye is pH sensitive, which means that you have this dye that lights up not only if there's calcium, but if the system is acidic, it's going to light up for that as well. And so if I take this dye and put it in the cytosol, yes, I can look at calcium, there's no problem. But if I put it in the lysosome, I don't know what I'm looking at, whether it's pH or whether it's calcium. And so what we said was, well, if you, and so whenever biologists used to look at acidic compartments and had to measure calcium, they'll run away from that problem. And so what we did was we turned the problem on its head. And we said, well, in this case, this dye is going to be sensitive. It's only sensitive to 2 things, right? So if we can measure the second thing, then we can introduce a correction factor. So if you take a duplex that's carrying this calcium sensitive dye and a calcium insensitive dye, this ratio is going to be sensitive to both pH as well as calcium. Now, if you attach onto this a pH sensor, such that in every lysosome, I know exactly what the pH value is. Then, if I know exactly how this dye's properties are going to change as a function of every single value of pH and calcium, then if I know the value of pH, I can introduce a pH correction factor and get the value of calcium. That's exactly what we did. So we showed that you can make this pH sensor, you can attach a calcium sensitive dye. This was synthesized by Nagarjun, who basically showed that you can get, you can see that how the calcium sensitive, the calcium sensing capacity, the KD, the affinity, changes as a function of pH, and you can describe that by an equation. And you can also see that using green and red we can tell calcium, using orange and red you can tell pH and calcium. And so I'm going to add a second layer onto this. So if you take your coelomocyte, which is now labeled with your device, you will take 4 images: the donor channel, the acceptor channel, the orange channel, and the red channel. This, I already told you how to get a pH map. This is going to be pH and calcium. And so what we were able to show using this was that you could measure the performance of the device, not just inside a cuvette. You could take it at every condition of pH and calcium inside a worm. And you can plot it. And this was done by a very talented graduate student, Anand, who was from IIT Guwahati. He's just finished his BTech and he's now a 4th year grad student. And this was all done by Anand. So I showed you how to get this D by A map, right? We already know you can describe this D by A as a function of pH by an equation. You can substitute the value of using this at every single, you can convert this D by A map into a pH map, right? We already know how pH changes as a function of the dye binding affinity. I showed you that map earlier. And so you can convert this pH map into a KD map, right? And now I have the O by R map, which is pH and calcium, right? And so if you multiply these two, using this equation that describes exactly how this changes, you can basically get exclusively the calcium map. I'm taking a little bit of time to tell you this because these are the first acidic calcium maps in history. And the reason I want to take a little bit of time is to tell you that this pH sensitive, pH sensing dyes have been known for about 50 years. Calcium sensitive dyes have been known for 40 years. The concept of endocytic tracers have been known for even longer than that, 40, 45 years. But we could not put all of these things together, right? It took a simple DNA duplex, which can act like an endocytic tracer, and take each of these components, put them on a 1:1 ratio to make a precise device that allows you to make a measure and be able to get a value of calcium. And so then what we were able to show was we could use this device and look at the calcium values in each as a function of endosomal maturation. So what we found was, of course, pH became more and more acidic as the system was undergoing endosomal maturation. This is nothing new, we know this. But this is new. Calcium values are fairly low in the early endosome and late endosome. Suddenly in the lysosome, there's a 35-fold increase in calcium. And so immediately we asked, who is bringing in calcium, right? And it turns out that we went and said, okay, who are all the lysosomal calcium importers that were known? We searched for weeks, we found nothing. We found large numbers of proteins that released calcium, right? So we know people who are releasing calcium. Somebody is putting calcium inside the lysosome, who is that? And so we then found that there was one report back in 1985 which said that we must have lysosomal calcium importers, but no genes were mentioned, no candidates were mentioned. So we know nothing about lysosomal calcium importers other than the fact that we definitely have them. Okay, and so what we did was we just asked, Well, we know who is the best calcium importer that's present on another organelle called the endoplasmic reticulum. And we know the lysosomal proteome. We know every single protein on the lysosome. This is public knowledge. So we went to the public database and we said, which of these proteins looks exactly like this importer called SERCA, right? And we found 8 candidates, and I really liked this guy, ATP13A2, because it looked so similar. So we said, okay, let's look at whether ATP13A2 is going to function as the importer. And so here's the system. The system we are going to look at is a certain protein called CUP5, the human homologous TRPML1. This is a very well-known calcium releaser. If it opens, calcium rushes out. Very, very well defined. And so what I'm going to show you are a series of experiments that will say that this protein CATP6, in C. elegans this is called CATP6, this protein CATP6 performs the exact opposite function of CUP5. So if CUP5 is releasing, this guy is going to be pulling in calcium. And how we're going to do it is the following. We're going to use this in the worm, and you're going to see 3 different phenotypes or 3 different properties, physical properties. One is, if you take, your worm is either going to be dead or alive. So if CUP5, this gene is mutated in the worm, both alleles, that worm is dead. If you somehow compromise the function of CUP5 in the worm, you're going to see a second phenotype, a second physical property. That is, this is going to be a subcellular phenotype. Your lysosomes are either going to be a normal size, or because you've created a lysosomal defect, you can't degrade cargo, boom, it just becomes like a balloon. And the third is the value of calcium. If you block a calcium releaser, so that's like a pressure cooker that cannot release calcium, right? If you plug it, then the value of calcium is going to become very high. And so what we were able to show was the following, that so CUP5 is a leaker, right? So if you delete it, the worm is dead, you have huge lysosomes, and you have high calcium. If you delete the accumulator as well in the background of this, it's going to set right this. Instead of having dead worms, you will have live worms. These worms will have normal sized lysosomes, and the calcium values are going to come back to normal. And so what we showed was that your worms were alive. We showed that instead of huge lysosomes now, the lysosomes came back to a normal size. And we were also able to show that if you delete a leaker, calcium values increase. If you delete an accumulator, which is this CATP6, calcium values plummet. If you delete both, calcium values come back to normal. And so at 3 levels, this gene is doing the exact opposite function of CUP5, and that is it's facilitating import. And so now the question is, what is this ATP13A2? It's also called PARC9, and it happens to be one of the major risk genes for Parkinson's disease. And so what we did was to ask, does it actually do this job in humans? And so we took normal humans' lysosomes and measured calcium. We got a certain value, 300 or 400 micromolar. And then we took skin biopsies from Kufor-Rakib syndrome patients. Kufor-Rakib syndrome is very sad. These patients are male patients who have very severe form of Parkinson's disease. It's early onset. They're just 35 years old or something. They basically have a single mutation, and the single mutation results in the ATP13A2 not being able to leave the ER. So it's stuck in the endoplasmic reticulum. As a result, the body is, the cells are making ATP13A2, they're just not on the lysosome, right? So there's, that's why these patients are ill. And if you measure calcium there, it's completely, it's down 12-fold, which basically means that ATP13A2's job is to bring calcium into the lysosome. People have been wanting to understand what this protein does. There are meetings that are held on this protein, but it was quite interesting for us to find that it has this function of importing lysosomal calcium.
And this is like some sort of an active transport that
It is an ATPase. It's a calcium ATPase, right? It's a P-type ATPase. And so the reason why, so why is it we who are chemists have found out the first lysosomal calcium importer? And the reason is the following, right? That if you want to look at a leaker, I put my calcium dye outside a neutral environment, so I can pop this balloon and see how much calcium comes out. So it's easy to investigate leakers. But if I want to look at importers, my probe has to be inside, right? And I can't do that. So now, since we can do that, now we can start looking at leakers. And so we want to make this probe widely available to all people who are studying lysosomal calcium releasers, because I don't know if you noticed, but for every lysosomal calcium releaser that's known, mutations in these causes a different neurodegenerative disease. And so it's highly unlikely that you would have so many releasers and just one importer. So now what our lab is doing is to try and see if we can identify new importers. But we want to make the probe widely available to everyone. And so with this, I'm going to just close and say that now with the capacity to measure chemicals inside live cells, one can actually start looking at the responsivity of cells to drugs, which I talked to you about. But also you can use it for discovery biology, which is, I think, important. Fundamental research, I think, drives all application. And I just spoke to you about the capacity of this technology to be used for personalized medicine, or rather precision medicine. We didn't say it, some editorial said it, and so that's why I put it here. And very recently, which I've not been able to talk to you about, is that we've now been able to look at, measure enzyme cleavage. All these ions at the end of the day, ion fluctuations result in enzymes not being able to act. And so now we have the capacity to measure enzymatic cleavage inside these organelles. This I didn't have time to talk about. It's just come out this month in Nature Nanotech. But I want to end by quoting Lord Kelvin, who says, when you can measure what you're speaking about and express it in numbers, you know something about it. When you cannot express it in numbers, your knowledge is of a meager and unsatisfactory kind. It may be the beginning of knowledge, but you have scarcely, in your thoughts, advanced to a stage of science. And so with that, I just want to say, I want to thank my wonderful group. Actually, all of this work was started by one very brave PhD student who is now a postdoc sitting in this audience somewhere here. His name is Shovik Modi. He's now working with Sandhya. I'm sure he shrunk inside his chair and I cannot see where he is. But it was a time when people were thinking that we were crazy to take DNA and measure ions inside cells. But I don't know where you are, Shovik, but I hope you can see what your work has grown into. And so all of this work was started by, I've acknowledged most people along the way, but the lab really is a mix of people from all areas. We have a chemical engineer, photophysicist, photochemist, a polymer chemist, biochemist, biotechnologist, geneticist. We collaborate heavily. For this particular work, we collaborated with Dimitri Krench, who gave us Parkinson's patient cell line. But most of the work was actually done in-house. And I thank these organizations for their funding, TIFR for believing in my vision and hiring me 12 years ago at NCBS, and I'm happy to take any questions. Yes.
So for your device sensing strategies,
Yes.
Using these aftermarket devices.
Yes.
So I assume for different strategies, the aftermarket designs are different, like nucleic acid.
Yes. Yes.
So my question is, how do you select the different kinds of different aftermarket for different strategies? So which strategies?
Yes. So I've been asked to repeat the question. So the question is, if you want to sense different analytes, how do you decide which aptamer to use? So what I mentioned, what I show you here, each sensor takes the rate determining step is actually sensor development, which we take for granted. And so it's really by trial and error. So either you can develop an aptamer by yourself, which is the long and painful way to do it, or you can go to the literature and you can see aptamers that already exist. You can do an affinity maturation and increase the tightness of binding of that aptamer, or if it's too tight, you can make single nucleotide mutations and change the KD of the aptamer. And then you do in vitro measurements to understand whether it has the sensitivity or the required KD for you before you make a sensor. Yes.
It was, of course, a fascinating talk.
Thank you.
I have two related questions. One is, what about some kind of calibration? I mean, do you have some way to find out that what the actual concentration that you're measuring is really very close to the real
Yes.
concentration?
Yes.
And secondly, how sort of sensitive are you to the environment, in the sense that are there some ions which if present will make your sensitivity less?
Can they interfere? Yes. So the first question is, do you do any calibration, or are you pulling these numbers out of thin air? And the second, so which I will answer first. So actually what I didn't show you is very rigorous calibration procedures, which are not just done in vitro, they're also done in vivo. I don't know if I have a slide for calibration. One second, give me a minute. Yeah, okay. Oh, okay, it's doing that. One second. Yes, so this is a very data heavy slide, which is why I skipped it. But it's just to tell you that you can look at the device, you can assess device performance quantitatively if it's a binding based sensor from these two parameters. One is the fold change in signal, and the other is the half point, the point at which you get half the response. You can do this in vitro, and then what you also do is you perform a similar experiment in the worm. You take the worm and you can clamp the ion concentration in the worm using what's called a clamping buffer, which sets the value of ions inside to a known value of the buffer you've added outside. And then you take a look at the sensor's response. And then you can compare. In vitro is in pink and in vivo is in gray. And so then you can actually quantitatively compare the performance of your thing. And now your sensor is actually sitting in vivo. So whatever is going to interfere should have interfered. But if it's giving you the exact same performance profile, then you know that it's sensitive to your ion of interest. And the second question was, I forgot.
How sensitive is it then?
I answered that, yeah.
And there was calibration here already.
Yes, yeah. Yes. Yes, Shamun.
Yeah, you can score. Oh.
It's already answered. Yeah, I answered both his questions.
No, because, sorry.
I have a question for you asking.
Yeah, because Roop has.
Oh.
Go ahead. No, most of the thing is the chloride that we've been talking
Yes,
and the calcium.
yes,
And the potassium.
yes.
So what are the counter ions for the calcium and the chloride? When you down-regulate or up-regulate chloride,
Yes.
what is the counter ion? Most of the calcium perhaps exists as a calcium chloride.
So in fact, people used to think of chloride as nothing but a counterbalance for protons or for calcium. So that is what sets the membrane potential inside this bag. You have chloride concentration on the outside, which is different from chloride concentration inside. Same for calcium, for potassium, for protons, and that sets your membrane potential. So as you know, yes.
When you have huge 6 orders of magnitude difference between the outside and inside,
Yes.
is that
So on the plasma membrane, so in fact I was going to, the talk that I would have given would have directly addressed membrane potential, because we've just been able to make a membrane potential sensor, and we've been able to measure membrane potential at the plasma membrane, which people have measured already, but we've also been able to measure the membrane potential of every single organelle inside the cell. And people didn't know what these membrane potentials were. Turns out that the lysosome has a membrane potential of plus 110 millivolts. The plasma membrane has a potential of minus 70 or minus 50 millivolts, depending on the cell line. But many of these organelles have membrane potentials of anywhere between plus 130 to plus 100 plus 130, plus 140 millivolts.
Negative inside?
Yes, so here's the interesting thing. It's actually supposed to be negative, but the convention for organelles is reversed. So they change it to plus, because that's how they insert their electrodes. Yeah, so that's the.
Just kind of a continuation
Yes.
of that. Could some of your calcium importers,
Yes.
if you want to find
Yes.
be using the proton gradient actually?
Yes, so here's the thing. We are planning to do this screen for C. elegans. So if there are C. elegans geneticists here who want to make a big splash, contact me later. But we have a capacity to identify now every single gene that will lead to lysosomal calcium accumulation. Some of these could be ATPases. In fact, ATP13A2 is a calcium ATPase. Some of them could be associating with a protein that is bringing in calcium. Like for example, CLH6 has this accessory factor OSTM1. So if it doesn't have the accessory factor, it's not going to work. And finally, some of these could actually be taking the organelle to the plasma membrane and then activating a crack channel to then achieve calcium uptake. So there are any number of mechanisms that one can think about.
Ankur.
See, we beautifully see this pH and this chloride, right? Do you see a third pair like HOCl crop up now in the lysosome?
Yeah, so we have, what I didn't talk about was our recent paper in Nature Chemical Biology that's ASAP, it's on ASAP, where we've shown that we can measure HOCl, which is the first example of measuring a reactive species inside an organelle called the phagosome. And in fact, a direct measure of HOCl can be made. And what we find is there's a very slow buildup of HOCl once you have the phagosome. But the moment the lysosome fuses, there's a huge jump, there's a burst of HOCl that comes about. And what we think is the following, that myeloperoxidase is already maybe present, right? But then it's also the delivery of a huge amount of chloride into the phagosome upon the fusion of the lysosome. That gives you the substrate chloride for myeloperoxidase to make HOCl. Myeloperoxidase, you can have some amount of myeloperoxidase that might be present. But actually when the lysosome fuses, it will deliver both myeloperoxidase as well as chloride, right? But everything else that the system needs is already there in the phagosome. So now you have fire and oxygen and yeah.
Yes. Beautiful talk and excellent work that you have brought into this particular field. So I would like to know how much far are we away from actually monitoring different ions in both space
Yes.
and in time across the entire micron length cellular envelope. So,
Yes.
so you have shown that this can be done in micromolar to millimolar concentrations.
Yes.
Can you go down to nanomolars and all the way up to millimolars in time? So how far are we away with these kinds of tools?
So what is the kind of change that what is the resolution that we are able to see? I think that is going to be determined by microscopy,
Yes.
right? Because I think chemicals are going to set, will set the threshold of where you can image and what quantity you can look at. After that, when you're looking at resolution, it is going to be dependent on microscopy and optics. Yeah.
Okay.
Yeah.
Okay, I think there are more questions. Of course, you can always interact with the speaker at the table, but we want to leave the speaker representing this nice.
Thank you.